Okay, let's just talk about vaccinations, which is the same as immunizations, right? So, what is a vaccination? Well, the vaccination is intended to uh, stimulate an adaptive immune response and have long-term memory. So, some vaccines use a live form of the infection, but it's attenuated, meaning it's disabled in some way. It can never give you the actual disease that it's trying to prevent. So it's live attenuated. And we typically don't give live vaccines to pregnant women or someone who's immunosuppressed. Okay, vaccines can also be dead bacterial extracts, meaning it's just a whole mishmash of the dead bacteria that's been uh, put in a vaccine and now injected into a patient, and the patient produces an adaptive immune system to all the bacterial components. Or a vaccine can be from the toxin that the bacteria produces. And tetanus is a perfect example of this, where we're giving immunizations to the toxin of the bacteria. And that's why we call it tetanus toxoid, right? So we're protecting you from the toxin that the bacteria is going to release. Okay, so the injection of tetanus is actually an injection of tetanus toxoid. Now, live vaccines are typically to viruses. So, for example, mumps, measles, rubella, chickenpox. And these are the ones that we avoid in patients that have reduced immunity, say they're receiving chemotherapy, or they're pregnant, because we do consider pregnancy a condition of immunosuppression. Okay. The vaccine stimulates the adaptive immunity, the B and T cells, towards that specific infection. And as I mentioned before, toxoid vaccinations, they're just made from the inactive toxin that the bacteria can produce if you were to get infected, and you make antibodies to the toxin. So if the body ever saw that toxin in the future, such as getting infected with tetanus, that then puts this toxin into your bloodstream, you would be protected from that. Okay, so here's just some examples of the different types of vaccines we use. And notice the live attenuated. Most of those are viral, except for tuberculosis. That's not a viral infection. But polio, measles, rotavirus, yellow fever, chickenpox could be in there. Those are viral infections. Inactivated, meaning we've taken uh, the bacteria or the virus and we've killed it. We've just inactivated it completely. Okay. Subunits means it's just been chopped up, and the subunits are now being put into a vaccine to be injected into patients. And then, of course, the toxoid. We mentioned that that's not the bacteria at all. It's just what it secretes into the bloodstream when you develop an infection. So, tetanus toxoid or diphtheria toxoid. So these are just all the different types of ways of making vaccines. Okay, let's talk about a few different definitions, and they'll apply to antibodies, right? So, what's an antibody titer? Well, an antibody titer is really a measurement in someone's bloodstream of the level of antibody they have to a certain infection that they've either come in contact with or they've had a vaccination to. Okay, so for example, we could give someone a series of hepatitis B vaccinations, and following the vaccination series, we could do a hepatitis B antibody titer. And that would tell us the degree of immunity the patient has to hepatitis B. So low serum titers typically indicate a lack of immunity. High serum titers would indicate good immunity to that specific infection. Okay, and we do check that in patients that have received certain vaccinations. Okay, what's an antibody screen? Well, when we're going to receive blood, the lab first takes blood from us to see what blood type we are. But they also take blood to determine if we have any strange antibodies in our bloodstream that may react to the blood that we're going to receive. And that's just called an antibody screen. Okay? And the same thing could happen to a pregnant woman. So a pregnant woman can develop antibodies to Rh factor, which is on the fetus's red blood cells. So we can actually do an antibody screen to see if the pregnant woman has Rh antibodies in her bloodstream that may damage and destroy the fetal red blood cells upon delivery or during labor. Okay, so that's an antibody screen. If you didn't understand the Rh factor part, we can discuss it at a tutorial, or I will bring it up again because it's going to be repeated on another slide. What's the amnestic antibody response versus the primary antibody response? So the primary antibody response is really the first time that the body sees an infection. The antibody response from the B cells is going to be mainly IgM antibodies. What's the amnestic response? Well, that's the future responses. The second, third, or fourth time that the body sees the same infection, it's actually going to produce IgG antibodies, which are the long-term antibodies produced by B cells. And we call that the amnestic response. The rapid increase in IgG because it has now a memory to that specific infection. Okay, so here's a lovely slide showing the initial response to an infection with the antibodies IgM and IgG. And you can see with the primary response, there's a big bump in IgM to begin with, and then the B cells start producing IgG weeks later. Okay. Now let's say months or years go by, and you have another potential infection with the same virus or bacteria. You still get a tiny IgM response, but look at the IgG response. And this is what we call the amnestic response, right? Or secondary response, because the immune system has already formed that adaptive humoral response. Okay, so really your long-term lasting antibodies are your IgG antibodies. Okay, so let's talk about the body's ability to recognize itself from foreign invaders. And really, it's the innate and adaptive immune system that are working together to recognize foreign invaders, such as bacteria or viruses, from cells that belong in the body, right? Their own cells. So the key to the immune system's response in recognizing itself is what we call the major histocompatibility complex. And this is just a set of genes that produces proteins that land on all of the cell's surfaces. Okay, so in humans, it's called the HLA system. Okay, so if you see MHC, which is major histocompatibility complex, or HLA, which is human leukocyte antigens, these are proteins that are on cells that allow the immune system to recognize itself, but also it can recognize foreign invaders. Okay? So let's say a foreign bacteria comes in and is attacked by a macrophage and then introduced to the T cells. Well, the macrophage will have antigens that are showing up on its surface from this particular bacteria, and the T cells can recognize it as foreign and destroy it. Okay? Compared to the proteins that are on all of your body's cells that it won't attack normally, because it recognizes them as self proteins. So all of the cells in the body have HLA proteins on the cell membrane, and the adaptive immune system is not supposed to attack them under normal circumstances. If you have an autoimmune disease, that will change things. Then you can have the immune system attack your own cells. Okay, so here's just an image of the HLA complex on cells. And this allows the immune system to recognize itself and not attack those cells, versus having antigens on the bacterial surface or cells that are infected with a virus. They will have different proteins on the surface, and the immune system can then hopefully recognize the non-self proteins and attack it. So again, a really amazing system of recognizing its own self. Okay, so let's just look at this protein a bit more, this HLA protein that's allowing the immune system to recognize itself and not attack it. So the HLA protein is on cell membranes throughout your body, and when it's on certain white blood cells, such as macrophages, they can present the foreign microbes or the antigens to the T cells. The T lymphocytes are then activated if the HLA molecule is presenting a foreign antigen or a microbe with it. Okay, and really this is the way that T cells recognize foreign invaders. This is also the way that T cells attack organs that have been transplanted, or tissue that's been transplanted. Is because the HLA antigens or proteins are different, and that's why we have to use long-term immunosuppressants to stop the T cells from invading organ transplants and destroying them. Okay, so it's a really important part of why the T cells and the immune system doesn't destroy itself. Okay, and everybody has slightly different HLAs on their cells, right? So only twins, identical twins, would have identical HLA proteins. And this is the major reason why immunosuppressants are required after organ transplantation. Even if you had a very close match, you would still need to be on an immunosuppressant. The only reason ever that you would never need to be on an immunosuppressant after an organ transplantation is if you received an organ from your identical twin. Okay, so let's look at this slide in terms of how the HLA system, or this MHC, the major histocompatibility complex, behaves. So the antigen presenting cell, let's just call it a macrophage, presents the foreign invader or the antigen with the HLA complex. and it presents it to the T cells, such as the T helper cells or the cytotoxic T cells. And then they mature and form a long-term memory to that specific antigen and also destroy the one that's present. Okay, so this can happen in the setting of a bacterial infection that comes into the bloodstream, or it could happen if someone didn't take their immunosuppressants and stop the T cells from invading a organ transplantation. Okay, so organ transplant rejections are really from T cells that are identifying the HLA complex on the donor organ and attacking those cells.